The main aim of the work package 1 of the DESIRA project has been to implement biotechnological methods for the identification and characterization of pests and diseases in Mauritius. Mauritius being a tropical and subtropical country, there are many pathogens, both viral, bacterial and fungal pathogens, that are causing a lot of damage to our crops. So the rationale behind this work package has been, or this approach has been, to use molecular techniques that are of higher resolution than conventional techniques for identification of microbial organisms. So conventional techniques like serotyping, biochemical characterization are very limited in what we can do. We cannot identify subspecies or strains of bacteria, for example. So this rationale has enabled us to implement this work package. So the first objective in order to start the work has been to upgrade our biotechnology lab at the Faculty of Agriculture of the university, and this was successfully done by the purchase of specific equipment that would allow us to use the molecular techniques. So why molecular techniques? As I've just said, we have better resolution in identifying both at the species level and at the subspecies level of any organism. So the first part of the project had been to target foodborne pathogens. So this was implemented by Dr. Huda Nitu. What she did was to test specific food produce, and we chose to focus on viruses because most of the work that has been done in Mauritius has been on bacterial pathogens. So we chose, and there were no reports so far on viruses that are found on food produce. So what we did was to test a number of food produce for both the noroviruses. So noroviruses is a large group of viruses which is reported around the world on food produce, and also for hepatitis A virus. So both of these viruses are actually RNA viruses. So they are a little bit more, it's a little bit more tricky to isolate the RNA in the lab and to do identification efficiently. But there were some produced in which we found both the noroviruses and the hepatitis A virus. The other main part of the project has been to focus on plant pathogens, mostly fungal pathogens. Two main groups of fungal pathogens in Mauritius are the Colletotrichum, in which there are many species, and each species targets different crops. So the aim here was to be able to use the molecular techniques to identify which species are actually on which crops, mainly chilies, bananas, and macadamia, and papayas as well. The other main group of fungal pathogens we worked on were the Stemphilium leaf blast, which is a major problem on onions. Okay, so what was the approach? Again, it was a conventional approach of DNA barcoding, which is used widely in the world. Why barcoding? Because it is a strategy for sequencing standardized regions of the genome, standardized region of the DNA of the organisms. So they are conventional regions that have been approved worldwide for which we can target the sequencing. So they are primers specifically designed that will isolate, for example, for animals, the target is a cytochrome oxidase subunit 1. For animals, this has been shown to be efficient, whereas for plants, other genes are used. They are plastid genes like the MAPK, the RBCL gene, in which we use the primers to amplify the genes, and then we do the sequencing. So these are accepted methods around the world that are being implemented in this work package. We're currently now working also on identification of insect pests, which cause diseases in Mauritius as well. They vehicle viruses and they cause a lot of damage to crops. So it's the same approach again, using barcoding of insects, but then the gene that is used for insect is different than for plants or for other organisms. So with this approach of using molecular techniques, it is far more accurate and efficient to be able to identify which species of pathogens we have, because this is critical in the management of the disease, and also to be able to identify are there different strains of the same species that are being vehicled around in the country. So we hope that with these the outcome of these projects would have been able then to help the agricultural sector in better managing diseases.